No, please, there's no sound. I'm not hearing anything. Have we started?
Good evening. No, we're starting
Starts at
at
7:30.
7:30. Yes.
Good evening, all.
Hello. Hello.
Hello, I can hear you.
Oh,
Evening.
yeah, good
Hello,
evening,
hello, we can hear
everyone.
you.
Good
I
evening,
can hear
everyone.
you.
Um, I
Good
want to,
evening.
good evening. I want to welcome everyone to the continuation of our clinical training course. Um, Please ensure that you fill the attendance, which is being posted on the link. Please, is important. And I want to, without much ado, I want to welcome the director of Ancestor, Professor Nodu, to kick off the meeting. Thank you. Hello, Professor Nadu.
Second, Professor Maxwell, please go on.
Okay, so once again, good evening, everyone. Just a quick recap before we start. This training is weekly on Thursdays, the same time for the next 10 sessions till the end of the course. 7:30 West African time, that's Nigerian time, so that we keep that in mind. We'll try as much as possible to send people reminders. And then at this point, just to let us know that we're going to have a quick pre-test now. Please ensure that you do the pre-test. Please, it's just we should be done basically in the next 3 minutes or 5 minutes at most with the pre-test so that we can start the first presentation. Just to please plead that today's session may be extended a little bit than the statutory 1.5 hours that is for every of the session, the weekly sessions. These sessions are 1.5 hours, but today there may be some slight, a few minutes beyond that, and that's because we want to take a topic that we didn't take during the launch last week, and that has to do with the history of clinical trials. So that will be the very last presentation. So please, let's quickly fill out the pre-test, as well as ensure that we have filled out our attendance links. And I want to urge that Dr. Maxwell Anyango from IQVIA get ready. And please, the team, the data team should please look out for him and make him a co-host so that he'll be able to speak. I think the slides are already with us, so we'll be changing it for him. Thank you.
Professor Maxwell is a speaker here. Dr. Patrick.
Oh, yes, let's search out. Mr. Ruben, can you see him? Let's just search the...
I can't find him, sir.
Do you mind if I have a presentation?
No, you can. Yes, you can go ahead. We don't mind.
Sorry, I'm here. I'm here. Dr. Akisheta is here.
Sure. Okay. Professor Maxwell, over to you.
Sir, it's the first person we have on the list is from IQVIA. That is Maxwell Anyango, who will be talking about clinical trial documentation. So if he's not here, we would just go ahead with the rest of the people who are here. I don't know, is there any IQVIA team? I know Dr. Kwaku was logged in. I don't know if you can provide any. I can't seem to see him, except if he's using another name. Please, is Dr. Maxwell Anyango here? Okay, so we definitely will have to move on with the other presentations. I hope we are, at this point, we should be done with our pre-test. If we are not, because of time management, we'll have to proceed with the presentations. People can continue to fill out the so the next, I think the best thing is that we should just go the way it is sequentially. The next is a recorded presentation. This session is 15 minutes. So the next is Dr. Nnamdi Okeke. So it's a recorded presentation, and please, the IT unit, can you put up Dr. Nnamdi's presentation? Please go ahead. You can share presentation.
That by the end of my presentation, you get to understand why we document in clinical trial, what the delegation of duty log looks like and why it's important, role and specific responsibility and communication flow, as well as also site-specific document handling and storage needs. So let's look at the bigger picture here. The average cost to bring a drug to market is over $1 billion, and it takes over 10 to 15 years to make this happen. Error in our data collection could jeopardize everything being worked for one particular drug in sickle cell disease or any disease. Incomplete data recording, as we've seen, duplicated data, inconsistency in data recording, outdated data or inaccurate data, all these could jeopardize $1 billion worth of research and all the science as well. And this is why data integrity is of importance in clinical research. Data refers to all recorded information that were generated during the conduct of a study, including and not limited to patient-reported outcomes or patient records, trial outcomes, safety findings, and regulatory documents. Data integrity is known as the bedrock of clinical trial. It ensures accuracy, completeness, and reliability of a data. Without data integrity, the scientific hypothesis regarding a drug will be invalid. Ethical questionable. Your data will be ethically questionable due to lack of data integrity. And then, of course, it's worthless in terms of regulatory processes. It will be of no use. So the importance of data integrity is to ensure patient safety, to ensure we're staying compliant with regulatory, and also for scientific validity, as well as also public trust. Now, the question is important that data integrity is incorporated in clinical research, but how do we go about this? Well, we can start with documentation and retention, because effective documentation and retention is an evidence of data integrity in research. If it is not documented, it never happened. This is why documentation is critical. The great thing about clinical research is everything you need, all the different guidelines you need to conduct an effective, ethical, and regulatory compliant research is already available at your footstep, right? We have the Good Documentation Practice Guideline, which is a cornerstone when you think about proper documentation in clinical research. So we'll look at what that is and then walk through that a little bit. What is good documentation practice? GDP, as you often call, refers to standard by which documents are created, reviewed, modified, maintained, and retained. It is also in compliance with some of the key ethical standards in clinical research, such as the International Council of Harmonization, GCP, regulatory compliance with NAFDAC, FDA, and EMA, and also sponsor requirements. Sponsors expect that every site conducting or running a study is in compliance with the GDP, the good documentation practice. What are some key principles to help you stay compliant within GDP? Here they are. So we often refer to this principle as the ACO Plus, where A stands for attributable, L legible, contemporaneous, O original, A accurate, C complete, and the C for consistent, enduring, and available. So in order for you to meet the principle of good documentation practice and maintain data integrity within your site, there is a need that these principles are upheld. And importance of data documentation is as follows. We document so that we can be able to reconstruct a trial at any point in time. By leveraging the data that were documented through clinical research, we can go back retrospectively to draw insights, to avoid patient safety, or to understand progression of disease and much more. Audit trail. Audit trail helps us to track what changes were made, why they were changed, and who did what. So documentation helps us to accomplish such. Regulatory scrutiny. Often you have site inspections by different regulatory bodies depending on where you are. Documentation also enables us to be site ready at any proper time. Proof of compliance. How do we make sure that every site is staying compliant with study protocol, GCP guidelines, and other regulatory and ethical requirements? This is also obtained through proper documentation. Documentation and retention are key principles in maintaining data integrity. If documentation is the physical or digital manifestation of data, then retention is the legal requirement to preserve that documentation for a specific period of time to ensure data integrity and accessibility for future review. Now let's look at retention a little bit in depth. Retention, just like the name mentioned, is to secure storage of trial documentation after the completion of a study. For ethical reasons, it ensures verification and audit readiness, helps with legal protection. Let's assume, not that we're hoping this happens, but there's a situation where there's a need to track back into a data to see what happened, right? So it protects every site from litigation or any product liability cases. History record maintains product development history for future research, future insights, future submission for comprehensive coverage from the regulatory point of view. Now the question is, how long should all these documents, all this data, all this information be retained for? It varies per regulatory authority. The FDA, the EMA, the NAFDAC, both all may have their own regulatory requirements. Retention period. So there's a strong need to make sure that for every study you are conducting or playing a key role in, you understand what the retention period are. Often, the retention period are often enforced by the sponsors, right? But the number that often jumps to mind is 25 plus years. We've seen the importance of documentation. We've seen the importance of retention. Now let's look at the clinical study team that actually makes this possible. The clinical trial stakeholder, there are a lot of them. It's not just about the PI and the site research team. It's about the communication with sponsors. It's about the CRO. It's about IRB and the participant, the patient himself. How do we maintain a flow of communication? How do we maintain compliance and transparency across all the different stakeholders. This is why roles and responsibilities are important in clinical research. Roles and responsibilities. The flow on my right shows the different oversights for a PI, and this is because the PI is fully responsible for overall trial conduct. Yes, as we can see, there are different pathways and different stakeholders that the PI has got to manage in order for trial to be successful. The sub-investigators have specific delegated duties they have to perform. The coordinators, they help with daily management and documentation. So if a coordinator is calling the PI frequently to collect certain information, to reconcile certain correspondences, please, they're just doing their job. Let's work with them to make sure we give them what they need. Question is, how do you ensure a clear flow of information and communication between the PI, the staff, the sponsor, the research nurse, the pharmacist, and many more? This is why we have various SOPs, delegation logs, and training logs to help define accountability so that we know who to reach to when it comes to communication, and that way maintaining communication integrity and driving clinical forward effectively. So we've looked at the PI responsibility. In research, there are different delegation log that helps PI to maintain proper oversight compliantly. One of those format documents is called delegation log. What does a delegation log look like? On my right hand side, we'll see what it looks like. So a delegation log is just a documentation of all the staff that has different tasks to perform through the life cycle of the study. Delegation log must be updated continuously as responsibilities change, and they are very important for accountability, data integrity, patient safety, transparency, and regulatory compliance. They come in different forms. So you may be used to hearing task delegation log is all the same thing. Anything to capture people's responsibility in conduction of clinical trial is what delegation log is. It's also a key document that must be handled, stored, and maintained as part of the investigator site's file. Now let's go to the site level and look at some of the important document handling and storage needs. So we've mentioned the investigator site file. It captures all the essential trial documents that must be stored to ensure regulatory compliance and inspection readiness. We are always making sure we are staying compliant regulatory-wise and also inspection readiness, both regulatory-wise or sponsor-wise, and many other bodies as well. Electronic system. Any digital record-keeping system must be compliant with the 21 CFR Part 11. This ensures that integrity, like I mentioned earlier, very important. Security, also auditability. Paper records, that is something that we are often comfortable when it comes to our sites in the sub-Saharan region of Africa. This being the case, it is important that the physical document must be stored securely, typically in a locked cabinet with restricted access, and remain readily accessible for audits and inspection. When an auditor or regulatory stakeholder approaches a site for inspection readiness and you're not able to provide your documents readily and easily, it shows that you're not being compliant. So this is important for us to maintain at the site level. Document retention, we looked at this per the ICHEC R3 guidelines, which is the most recent one. It is required that trial records be retained for at least 25 years post-study completion. But of course, again, please check with the region. Examples of documentation issues. So for instance, risk area could be regulatory. Issue could be missing of investor CV. In part, you're being non-compliant once that happens. So action required is speak with the investigator, get the document, update the CV, verify qualification. Always make sure qualified individuals are performing clinical trial activities and tasks. Quality assurance, you have a CAPA that is open. CAPA tracker has not been updated since its origination. That is a weak audit trail. So you have to measure, review your tracker, log actions, and follow up. So these are some of the other few things that some of examples that we can see with documentation issues. So in conclusion, the success of any clinical trial hangs on 4 fundamental practices: robust document management, clear delegation of responsibility, consistent, effective communication, site-specific handling of essential records. Every single record document is critical to drive clinical trial forward. Here are some references if you care to learn more about some of the things I've provided a little bit in depth. I want to thank you for this opportunity once more again, and I hope that I was able to shed some light on why documentation and retention are important, how to do it, and all the different guidelines that guide us in terms of maintaining and retaining documents, and also making sure we're staying regulatory compliant. Thank you once again, and have a great day all. Bye.
So we're done with the first presentation. Next, we'll be going to that by Dr. Shea Akinshete from Lagos University Teaching Hospital. He's going to be sharing experience on the clinical trials that he's been handling there. Please, admin, can we make Adeshe Akinshete? Okay, he's already a co-host. Please go ahead with your presentation.
Sorry, I think I got it wrong. I imagined I was to talk about delegation of duty log, but now I'm being told to talk about clinical trial experience in Lagos.
Oh, okay. Please just go ahead with your presentation.
Oh, no, I don't know,
you
because
forwarded
now the previous speaker has almost said quite a number of the things that I was to do.
It will
But
not
the
be
person
the
I
same
interacted
thing.
with talked about delegation.
No, Shay, just give your
Okay, ma.
presentation, please.
Yeah.
Thank you.
Okay, ma. Can you see my screen, please? So I'll just be talking. Can you see my screen?
We can, but it's not in presentation mode.
Yes,
Okay.
yes.
Okay. You can put it on presentation mode, please.
Yeah, I will do that. I'm going to do that. Yeah. Can you see it?
Yes, we can.
It's on slideshow now. So I'll be talking about delegation of duty and document management. And I will try to just relate it to the experience as you've shared now so that I can run through. So my name is Adeshe Akinshete. I'm a pediatric hematologist in Lagos University Teaching Hospital. So basically, documents, their management, their acquisition, management, storage, and retention are the key highlights of a clinical trial, because what it tends to do is that if somebody comes 5-10 years down the line and look at what was documented and try to run the analysis of the data sets, they should get something that is close to what was gotten when the trial was done. So the variance should be minimal. So delegation of duty, document management, regulatory compliance, and patient safety will be the key. Now, the critical person in a clinical trial is the principal investigator. The challenge with clinical trial is that most clinicians imagine that clinical trial is clinical practice. The two are very far from each other. So my experience was that the first time I got on a clinical trial, there were a lot of mishaps. There were a lot of wrong documentation. There were a lot of bad practices that were not GCP compliant. So a classic example, you see a patient and you write a scroll. That your scroll is not legible to the next person reading. In a clinical trial, that is unacceptable. In clinical practice, we get away with it with that lie. Oh, it's doctor's writing. There's nothing like doctor's writing. What you write must be ALCOA compliant. Somebody must be able to read it. Somebody must be able to understand it. It must be legible enough. It must be reproducible. So all of those things were things we had to learn. The other bit was that in clinical practice, you will say that, oh, you are the doctor. And so the nurse will be the nurse. The pharmacist is the pharmacist. The lab scientist is the lab scientist. That concept is somewhat what is retained in the delegations of duty logs or the delegation of authority. What it means is that the principal investigator has to, one, have the requisite education, two, the requisite training, and that person must be able to deliver what the sponsor requires as to that particular clinical trial. So what it means is that there is a track record to what you have been doing. And so the sponsor gives you a molecule. Now, the intricate nature is that the sponsor talks to the institution that I need a PI in large prints. However, the sponsor is talking to the PI, but every discussion or every conversation is done with the PI who is attached to the institution. Now, delegation of authority now means that the sponsor hands over a clinical trial. So let's say hypothetically, Panadol is going to be tested to see if when you give Panadol, you will reduce the amount of protein in the urine of people. So they identify me as a principal investigator. Now I need to have a team. That team needs to be people who can do the duties. So that's delegation of duties. So I look for a pharmacist who by nature has the training and the requisite skill set to administer that paracetamol, which is now an investigational product, to people. I get a nurse who has the training and skill set. I get a laboratory scientist who has the training and skill set. I get somebody who will be a study coordinator. And so all of those things are necessary. Now, in the delegation of duties, what it then entails is that the principal investigator retains overarching responsibility. So even though the pharmacist is the one in charge of dispensing, the principal investigator is liable for the errors of the pharmacist. Even though the nurse is the one who is taking temperatures and vital signs and pulse oximetry readings, the principal investigator has overall oversight for it. That's one arm of it. The other arm is that all the documents and the information generated throughout the trial will need to be overseen by the principal investigator. So you need to look at each of those documents at the end of the processes and determine that everything was fully signed up. So what are the critical things? It's a regulatory thing. You have regulators who say you must delegate responsibility and you must go ahead and oversee. So let me share before I understood delegation of duty. So when we had one of the trials we had, in my head, I had delegated the responsibilities to monitor temperature excursion with regards IP to the pharmacist. So that particular day, there was an excursion and sponsor reached out to ask, oh, there was this excursion. The mail was directed to the principal investigator, copying the pharmacist. Sponsor reached out, oh, there was an excursion. Can you give us the details of what was done? How did you go about it? Who have you reported to? What are your mitigants to this thing not happening again? And I called the CRA and said, look at this, look at this. This is what I want to respond. That pharmacist, and the CRA says, no, it's not the pharmacist who is in charge. You are the principal investigator. Kindly flip to your delegation of duty log and look at the opening statement there. says the principal investigator is delegating this duty to you. So you are still the critical person responsible. So that changed completely for me, that at the end of every week, as a principal investigator, I would look at the delegation of duty log. I will look at the people who are assigned. At the end of every quarter, we will look at what they have in terms of their certificates vis-a-vis what they are signed up for, so that somebody will not do a trial activity while their licenses have elapsed. That is a protocol deviation. So that's critical, that every principal investigator should understand that one of the most critical documents for them is the delegation of duty logs. There, only people who are signed up for tasks must perform those tasks. If somebody is not signed up for a task and they perform it, it becomes a protocol deviation. And some protocol deviations can be a huge problem. Let me give you an instance. You don't sign up somebody for informed consenting, and then that person, because you say is a doctor, goes ahead to consent a participant, and you go ahead to then do trial activities on that participant. That's a huge protocol deviation, because that set of activities is based on a faulty premise. There was nothing done, and you may get kicked out of a trial for that. So that's important for us to note. So I've put here the name of the person is important. They must sign the delegation of duty log. Now, this is important because the signature that is on your DOA or your DOD log must be the same signature that must be on all trial documents. So if somebody has signed OAP on your delegation of duty log, they are not permitted to sign APO or PAO. That will be flagged as an inconsistency, and it may make those documents not admissible for trial. So the way they have signed in the DOD, they must maintain that signature all through the trial. That is very critical for us. Every activity has a start date and it has an end date, and you must adhere to this. Once somebody leaves the trial, PI should sign them off and say their study has ended. The other thing is that as a PI, you can add roles. So I want to give a classic example. We call participants every week to find out how they are doing. There is no role on the DOD where you say you call participants. So you create it. So we said calling of participants, and we assigned it to someone. And that person is the one who does the role. So I can't then get a nurse who hasn't been assigned to call participants, calling participants and documenting. That's not that person's designated. So it's important that we do that. Now, these are, I've talked about qualification and kind. So let's go to documentation. The documents that most of our trials utilize are paper-based documentation. What we have tried to do is, when we started, one of the most critical documents is the informed consent form. We had our teething challenges with how we worded our consent form, with how we did our consent checklist and all. But now, we've gone through several cycles, so we know that informed consent, when you have finished the consenting, you must write in longhand everything you did in that conversation. The risk, the benefits, the molecule, everything is written out. All the questions you asked that person, you write it. And that document is quality checked by 2 people, apart from the person who got the consent. And that document is filed. Every time there is an amendment, you go through that process again and you filed. Because the basis for a sound trial, for data to be admissible, is informed consent documentation. Then you have your daily sheets. We have had to create our own SAE forms, AE forms. We've created sheets for every week of different trials because the assessments are different. And all of those things, they contribute to deviations. and they all have to do with documentation. So if you are not supposed to take PCV, the fundamental thing is that, oh, you are doing a sickle cell study, I will take PCV. That is a deviation. But when you have developed your document that for week 3, all the investigation for week 3 are on that sheet, then it's easy for everyone to follow through and not make mistakes. So the core documents that are in the trial master file are your protocol and the amendments. So the initial protocol and all the amendments. And as all these amendments are coming, you are going to your ethics committee, you are getting the approvals, you are filing them, you are amending. If consents were amended, you are retaking consent to be right. Your investigation brochure is a trial master file document that must be up to date. Informed consent, I said, your regulatory approvals, NREG, NAFDAC, as well as your institutional ethics board. The CVs and qualification documents, this is another area where we always have issues because CVs should be timelines of 2 years. So most times it falls through the crack. We don't remember. But because we now do quality checks by the PI, you look through. Let me look at the document. Let me look at everybody's CVs. Let me look at everybody's practicing license. Let me look at all the documents that we need so that we don't get somebody doing a task that their licensing has elapsed for. DOD or DOA, as I said, source documents, and then the CRFs and monitoring reports. I've talked about trial master files. So for us, we don't do the remote trial master files because we are not largely electronic. Most of our documents are in paper-based forms. We review them and we keep them in a fireproof cabinet to ensure that everything moves on smoothly. What is the essence of this delegation and documentation together? So it is precisely for an audit trail. When we think there is a problem, it is easy for us to link our DOD and look at who is the corresponding official who is supposed to get this document out, and then PI escalates. It is easy for us from an accountability standpoint, and most importantly, that we are complying with all instituted GCP and local regulatory authorities for that. So we don't do most of the modern technology things. We don't do them, but it's good for us to know because we're increasingly now getting trials where the sponsors are moving away from paper-based promise fatigue scale, 6-minute walk scale, and all those other things. They are moving to the patients have their devices and they are putting their data there, and we are now having to go electronically to review them. So hopefully, as we update systems, we're getting to a point where we may be completely electronic in our documentation. In conclusion, it's critical that we understand that the principal investigator has complete oversight for the clinical trial. He is the one that delegates responsibilities. He identifies the people and he delegates to them, and he continues to maintain that oversight. He is the person that everybody knows has to do that work. And it's advisable that every quarter, principal investigators should go through the delegation of duty logs to update all their staff sites who are there. It is also important that the PI should be conversant with the trial master file, which is the document that gives you your comfort zone and confidence that, look, we are going by the letter and we are compliant with all the things that need to be done. The caveat or the challenge we've had is that we all understand that we have professionals that are moving. So you train some professionals and in 2-3 years, they've moved on. So we have to keep training. The second thing is that we have to get people who are really motivated to work on a clinical trials team because it is not your usual work. It is an extra work that is demanding and you need to put in a lot of effort. So by and large, delegation of duty and good documentation go hand in hand. When we've delegated properly, it's easy to document properly, and it's easy to synergize them and ensure that we deliver a good trial. Thank you very much.
Thank you very much, Dr. Akinshete. Thank you so much. Um, this is still practical experience that you're sharing. So the introduction I gave is not out of place. The weekly sessions are in themes. So this week's theme is about documentation. So whatever your topic reads like, it still has to do with documentation. Thank you so much. Thank you so much. So we'll move ahead now. The next, we just learned that the Akivia person has been around. He was under a different name. So we'll be taking the first presentation that is supposed to be given by Dr. Maxwell Ayambu. And once we are done with that, then we'll take Professor Pupola. Thank you. So can the team share Dr. Ayambu's slides and unmute him? Please look for Frida, Dr. Frida. I think they're together, so that it's Dr. Frida that you unmute. IT team, please grant co-host to Dr. Frida. Okay.
Hello, everybody. So he tried to log off and log back in. So he is rejoining in a minute, and he might rejoin showing us my name because his link is not working. So let me just rush him to join. Give us a few seconds, please.
Hello, good evening, everyone.
Good evening.
Good evening. Good evening.
Thank you. You can hear me clearly?
Yes, we can. Please go ahead.
Thank you.
Good evening.
So let me start by introducing myself. My name is Maxwell Anyango. I am the Associate Director for Quality Management for Sub-Saharan African region, responsible for quality delivery, also supporting our sites and our site management teams in terms of quality delivery for the region. So it's my pleasure to be here with you. Sorry, I had some challenges to join in. So you can see my name there saying Frida, but that's fine because you can hear me. I think we should be able still to go ahead and deliver for the purposes of this session. So this evening, I will take you through the clinical trial documentation. So feel free to ask questions or post them in the chat, and then after my presentation, we should be able to discuss them. Okay, so we can move on to the next slide. So why is clinical trial documentations critical in clinical trials? So as you have seen perhaps in the earlier sessions before this, is a huge difference between clinical trials and clinical practice. And one hallmark of clinical trials is the amount of documentation that is involved. And usually this is before, during, and after the trial. So if you've been working in clinical trials for some time, you'll appreciate the amount of documents and the sheer volumes that are generated. Now, the amount and the type of records that will be generated before, during, and after the trial will depend on a number of things. So not two type of trials will have the same amount of documentations and so on. So one, the trial design, then the conduct and the procedures involved, and then So I can see in the chat there is a question that says we need a pretest. Okay.
Dr. Yango, just go ahead.
So I go ahead.
Staff
Okay.
yes, they will handle any of those things being posted.
Okay.
Yeah.
Thank you. Thank you for that then. And then now with the new direction that trials are taking on risk-based monitoring, also it will depend on what is the importance of those documents and so on. So the new GCP revision 3 has introduced a lot of flexibility around this. And then most importantly, the relevance of each record to the trial outcomes. So those 4 key considerations is what will determine if we are filing that particular documentation or not, and we're going to see the details in the next few slides. So we can move on to the next one. So what is the purpose for filing and for maintaining all these records? So there are key 4 functions for that. And we shall also look at who is responsible for that. Now, in general, all these documentations constitute what is called a trial master file. Now, from a regulatory perspective, a clinical trial only has one trial master file with 2 components, the sponsor component and the investigator component. So commonly in practice, we refer to the site component as
father
the
build house. That is
investigator
mistake.
site.
It will all Kanka house.
And then for the sponsor component, then traditionally we
If
refer
the
to the
father build them in Lagos.
trial master file.
No, the way you know about the look at property.
Please, you can mute
If they
yourself.
help me,
There's
give me
some
small.
So in muting that person, we may have muted the speaker. So can admin unmute the
Admin, please.
speaker now?
Okay, he's been unmuted.
Thank you.
Okay, can you hear me?
Yes, we can.
Thank you. Thank you. So essential records are documents that either singly or collectively allow for the reconstruction of how a clinical trial was conducted in order to independently verify that that trial was conducted as per protocol, and 2, it made the regulatory compliance, and 3, that the data that has been collected is reliable, accurate, and credible. And lastly, that the safety, the rights of the participants were safeguarded. So that is the main reason why we collect and file all these documentations to ensure that, or to support GCP and regulatory compliance, to ensure data reliability, and then also to demonstrate investigator and sponsor oversight, which is now something that is really being stressed upon. And then during the audits and inspections where this reconstruction is done, they're able to review all those documents collectively and now reconstruct those events for purposes of those audits and inspections. And at times also you can have our local IRBs conducting their own independent audits. So that is why it is critical that we file those documents as is required and properly to enable to support these purpose of reconstructing these events so that there is no gap in the reconstruction, because once there is a gap, then it means the story doesn't add up, and then the queries will be asked, and then that is usually where problems start stemming from. So who is responsible from a site perspective to maintain these records? It is the investigator or the institution. So I was listening into the session that just ended, and they were talking about the peers' responsibilities and so on. So the principal investigator primarily is responsible for maintaining those records, but he can delegate that function to either a study coordinator or any other site staff who has been trained to be able to conduct those purposes. So normally these documents are filed in what we call the investigator site file, and we shall look at what are the basic elements that constitute the investigator site file. But there are the basics that must be there. Once you have this file that is arranged in a particular order, one, you must always maintain access to these essential records at any given time. And why is that important? For some reasons, for example, you might not want to file certain documents within the file because of either the sensitivity of the nature of the information contained in those documents. So, for example, financial documentations of a clinical trial. So the PI might not want to file that with the regulatory binder. So that might be kept separately either in the finance office or in his office and so on. There must be documentation in the file to show where that is filed, and any other document for that matter that is not in the file for that at that particular time. So access is extremely important. One, to confirm that these documents actually exist, and two, to be able to assist the inspectors or the auditors to reconstruct those events. Then proper retention is another thing that is essentially, it's very critical, because we might file, but then each country has its own regulations as to what is the duration of time that those documents must be retained. So premature destruction of those documents must be prevented at all costs. And in terms of practicality, it means then that you must think about how are we going to file these records? Is it in a lockable cabinet? Is it in a place that is not accessible? Is it in a place that the records will not be damaged by the elements? And we're talking about humidity, we're talking about insects, we're talking about fire, we're talking about flooding and things like that. So even during inspections, those are some of the basic elements that they'll be checking to ensure that we retain those records for the minimum time as required, either by the sponsor or by the local regulatory requirements, depending on which one is longer. Okay, so we can move on to the next slide. Thank you. You've skipped one. We can go back. Okay, so then, thank you. So the essentiality of clinical trial records. So it's important to determine what document do we file and which one do we not file. So I've just given a basic framework that you can work around with, the 4 critical elements that will help you decide if this particular document that I'm looking at meets one of those criteria. But when you are coming up with that assessment criteria to decide what document is an essential document, then you need to have a criteria. And the important thing is that it's not a must that you need to document, the criteria that you use to determine you are filing this and not filing that. And this is a new development out of the ICH GCP revision 3. Previously in revision 2, they just had a long list of what needs to be filed before, what needs to be filed after, and so on. It's still a good list for a site to use if they have still not developed, the skill out of practice or out of experience. So that list still is very useful. So if you're not sure or if your site is new, you can always refer to revision 2 of ICH GCP. It has a list that just lists before trial, these are the documents you need to file in the investigator site file and in the sponsor's site file and so on. But with the current ICH GCP revision 3, it does not contain that table the way it was in revision 2, but rather it gives you a framework within which to operate with, which I think is very good because what that simply means is that there is no one size fits all, or rules, hard set rules to say that in every trial, this is what must be filed before and during and after, because depending on the clinical trial and all those other aspects that you have looked at, it might not be applicable for that particular study. And therefore, what that means that in terms of practice, it will help you to avoid all these many sections that you create in the investigator site file that you say not applicable for this trial, not applicable for this trial. And that is why those changes were made to bring that framework so that now you're able to decide, is this relevant for this trial or this is not relevant for this trial and so on. So what makes a record essential? That is what you need to ask yourself. So one, does it contribute to trial oversight? If the answer is yes, then it means you need to file it. Does it support our regulatory compliance? One, in terms of GCP compliance, in terms of the local regulator guidelines and conduct for clinical trials, or even the local IRB requirements. If the answer is yes, then it means we need to file those documents. And three, does it support or ensure data integrity and reliability? If the answer is yes, then it means we need to file it. So that will depend now in terms of the clinical trial design and the risk-based elements and so on. Then depending on what we are generating, then we'll know we need to file those ones. Unlike previously where we had a long list and it will say you need to have this and you need to have this and that and so on. So for instance, if a study is a non-interventional trial, so there is no investigational product, it simply means you do not need to create a section for investigational product in the investigator site file because it will not be applicable. And therefore it means when you be creating the table of contents, that section will not be there for a trial that does not have an intervention. Yeah, but in R2, those sections are all listed down and the whole had to be created, and then I will be having note to files explaining this is not applicable for the study and things like that, which creates unnecessary administrative work for those maintaining the investigator site file. So that is the critical difference or variance between revision 2 and revision 3 of ICH GCP that we are currently using. The next slide. So the next slide just gives us more details in terms of what is the criteria for essential records. Yeah. So one, regulatory and ethics documentation. So all the documentation we prepare in terms of seeking approvals from the local regulator and the ethics committees, those must be filed right from the initial submission letters to the rest to the comments that they write back to us and the response that we make to those comments and all that back and forth until the final decision is made, which is either an approval or a denial of it or conditional approval and things like that. So we need to file all that communication so that when somebody is reviewing it, they're able to see these are the documents that you submitted. And it's important that when we are crafting those submission letters, we include all the documents, including the document name, the version dates, and the version numbers in the cover letter so that now you're able to track. Because at times you might submit version 1, then the IRB or the regulator sends comments back and you update it to version 2, and then eventually what is approved is version 3. So if you do not track it to those details, then at the time of you filing the approval letter, somebody trying to reconstruct these events might not be able to know what was the actual version that was eventually approved. So it's important that even when you are drafting the cover letters, you include the details of what you're submitting so that once the approval letter comes, you are able to trace based on the back and forth communication to confirm that these are the right documentations. Now, in terms of trial conduct and oversight, you might have trial-specific plans, MOPs, and things like that, manual of operations, that is what it means, or even your SOPs and things like that, PI oversight plans. All this needs to be filed in the investigator site file. The PI, together with their site staff, might have regular meetings. It's important to file this because PIs are very busy people, and there is no expectation that they sit there in person every day, running trials and things like that, as long as they have a team that is trained and delegated properly, as you have had in the previous section. However, there is a requirement that we should be able to demonstrate that they have adequate oversight activities for the trial. And this includes their personal involvement and two, the, the, the ability to demonstrate that they are supervising those whom they have delegated trial significant activities. And meetings is one way that they demonstrate that. So if you have those trial meetings with the site staff on a regular basis, then they're minuted. It's important also to file those ones because that is one way you'll be able to demonstrate PI oversight activities. Then things like documentation of trial procedures, this could come in the form of clarification memos from the sponsor and things like that. We need to file all those because all these are ways in which we demonstrate trial conduct and oversight. So all those documents will constitute essential documents that need to be filed. Next slide. Then participant safety and consent, it's very critical. So that is another aspect that they will also look at. And we know in practice, most of the time, participant safety will be recorded in clinical trials in terms of how we are identifying and reporting adverse events and serious adverse events and any other reportable events based on the protocol. So that is something that will be looked at. So it's important that when you are creating templates to record this, one, it must meet the protocol criteria for collection of these events. And two, it must also meet the local regulatory criteria. And if there is a variance between the two, then it's always important to go with the most stringent one, which most of the time is the local regulatory requirement one. Then informed consent also is very critical because it forms the contract between the participant and the PI in terms of their participation into the study. And therefore, how we conduct that process is critical that you think about how we are going to document that. Because as you will hear, or you might have already heard, that informed consent is not just about signing that document. So it is a process. So then how do we review that process? How do we construct the events of that process? So it's important to have a template in place that will capture the key elements of that process. And that also needs to be filed. So usually it will be filed with part of the participant's file, or if the site's process is different, to file them separately. Then that process must be well documented. There is no one best way of doing it so long as it meets the threshold of ICH GCP and the local regulatory requirements. So that different sites do it differently. Then records of participants' existence and data integrity. So it's critical that when you are creating a file for a participant, you think about what will constitute source for each of the data points that a protocol requires, so that if a monitor comes or an inspector comes or an auditor for that matter, they're able to look at the totality of this documentation and confirm that this is a participant who really exists. He's a real person, and these documents are not just a collection of documents for an individual who cannot be confirmed to exist, or rather fabrication of data for that matter. So it is important, even when you're thinking about developing those source templates, as a site, you ask yourself, what will be the source? And the best practice is to develop one source template for one source data point so that you avoid the duplication, you minimize transcription, and that way, then you are able to meet the Alcoa principles well, and also to reduce the administrative task in terms of, transcribing information that has already been generated and can easily be entered into the electronic data capture. Then demonstration of qualification and delegation. So the previous session talked a lot about delegation log and its importance. So that again is another essential document that must be filed in the investigator site file. So I'll not talk too much about that because it has already been explained in details. Next slide. Then investigational product. So the caveat here is if the study involves an intervention. So you could be doing a clinical trial that does not involve an IP, or the interventions is not an IP, or maybe it is a device and things like that. So the important thing here is whatever the intervention is, whether a device, it is an IP, a vaccine, and so on, all the documentations surrounding the IP, the investigational product, from the time when each pill was sent from the manufacturer to the time it was consumed by the participant, we should be able to trace each of it. And it is a primary responsibility of the investigator to ensure that those investigational products are used as per protocol. So that traceability and accountability is critical from the time the manufacturer sends it or the sponsor sends it to the time it is destroyed. So when we'll be performing IP accountability, or the monitors will be doing that, that is what they'll be trying to look at. So you received 200 IP kits, you used 180, we expect to see 20 have remained, and who used the 180 and when. So if there's any gap anywhere, then those are the issues that will be looked at. If, for instance, there are certain temperature requirements that must be maintained, then they'll be looking at the cold chain and things like that. So it's essentially critical that the pharmacist knows how to document that properly so that it can be accounted for at the site level and then at the participant level. And usually the sponsor will provide the different kind of logs to perform these activities, and those must be maintained and kept current and up to date. Records of unblinding. So depending on the study design, some studies are single blind. So if a study is single blind, it means one team will know what their IP assignments are, and then there must be the other team which is blinded that must not know. So there must be a site blinding plan that delineates these are the unblinding team and these are the blinded team, and they must not cross. So that firewall must be maintained for purposes of maintaining the scientific integrity of the study, and those must be properly defined and filed so that during the reconstruction of these events and comparing it with how people are delegated and how the source documents are being filed, we can verify independently that that blinding plan is being followed and there are no risks or instances of unblinding of the study. Then another essential documents are systems and data integrity documentations. So if the site is utilizing any electronic systems, so this could either be sponsor provided, like the EDC systems, or they could be site owned. Yeah, so some sites owned electronic systems. A common one is like the laboratory management information systems. So they could have automated some activities at the lab, for instance, or it could be an electronic investigator site file. So usually if it is sponsor provided, then the validation documentation, they usually provide this. But if it is a site-owned system, then it's important that you ensure that the validation documentation is in place and it is filed in the investigator site file. Now, just a note to remember is that for sponsor-provided systems like the EDC systems and randomization systems and things like that, routinely, they do not share the validation documentations with the sites because as per ICGCP previously, this required to be kept in the sponsor component of the TMF. However, what we have noticed in Africa and in different countries, the local regulators, because many are using the AVRF checklist, and it has that as one of the things that they look at. So increasingly, they are asking for validation certificates, even for the EDC and the randomization systems and so on. And they catch the site off guard because traditionally it's not filed in the ISF routinely, and it's generating a lot of audit findings. So that is just something you should have at the back of your mind, so that should you be having an inspection, then you should be ready to request it from the sponsor, because most of the time they will provide it if you tell them that the local inspectors want to see it. So it's usually there. But if it's your own systems, then always ensure that those validation systems and data integrity security systems are there. Metadata and things like that, audit trails and things like that must always be there, because it's what we call Part 11 compliance, that all these electronic systems must always meet. Next slide. Then agreements between parties, CTAs, insurance, and things like that also need to be filed. Protocol compliance, how we are handling samples, and records of what we have collected and what we have shipped. Also, those all constitute essential documents. Next slide. So before the trial conduct, so what do we need to file? So usually, in general, this will be communication with the regulator, because we are still trying to obtain approvals. And then it will be also documents that the sponsor has generated in terms of enabling the site to conduct the trial, investigator brochure, the protocol, and things like that, informed consent process, and documents like that. So these are the bulk of the documents that will be collected and filed before the trial conduct. And then once approval is received, then for this kind of documents, if there are any that have an expiry period, then it will simply be a matter of applying for renewal. Yeah. Or in the things like investigator brochure that requires an update every year, then a new one will be sent, and then we just make a notification, and then we just update those sections. If there are any protocol amendments, then those will also be filed. So in general, that is what is filed before a trial. But if you go to ICGCP, there's a very long list, and I think I also created one in these slides that I will not go into details, but it's just to give you an idea of what this could be like. And then during the trial, what do we need to file? Next slide. So operational logistical, again, this is still before the trial, the trainings that we do before we are delegated and things like that, normal ranges and so on. Next slide. Then during the trial now, we start recruiting participants. That is when the bulk of participant-specific data starts coming in. So mostly you'll find participants' files keep on growing bigger and bigger. If there are any safety events, adverse events, and things like that, then that will be the bulk of what we'll be collecting during the trial conduct. Then monitoring follow-up letters, confirmation letters, if there are any audits and things like that, communication between sponsor and ECs, then those are now the sections that will also be completing as the study continues. Next slide. Then trial oversight, as I said, when you'll be conducting your meetings with the site staff and things like that, collecting samples, shipping them to central lab, or storing them at the site waiting for shipping towards the end. IP accountability, because we are now accounting for what was sent to us as the participants are taking. So we'll be filing all those during the trial conduct. Next slide. Then after the trial conduct, when most of these things have been done, so we are focusing on closing the study. So we'll be closing the delegation logs, ensuring that the biological sample trackers have been reconciled and the central lab has confirmed everything has been received, and then making the final notifications to the ethics that yes, you gave us this approval, you've already done the study, this is the final progress report. And once the CSR is received, we'll be able to notify you of this. Okay. So after the trial conduct, the amount of documentation is significantly smaller compared to the other two stages. Next slide. Yes, so then again, participants' identity. So the master identification log. So these are kept for they're kept safely, because of the potential identifiable information that they contained. And then if there's any unblinding documentation, so some protocols might require that participants are informed whether they received the IP or they received the placebo and things like that. So if that is part of the protocol, then also those are part of the documentations that will be filed. Next slide. So that's my final slide, just right on time. So if there are any questions, I would like to take any questions at this point.
So thank you very much, Dr. Yango. We will take questions at the end. So I want to appeal to you and the previous presenter, Dr. Shea, to just hold on. We have just two presentations that have to do with still under the theme of documentation, but from the registry aspect, one of those things that we need to be conscious of that is required as a basis to be sort of ready, preparedness. So please, let's welcome Professor Ademola Pupola to do his presentation. Prof. Please, can the team make Prof. Ademola Pupola? Okay, he's a co-host now. Please, Prof, go ahead. Please, can we unmute Prof? Prof, please, can you unmute yourself? You've been made a co-host, so you can share your slides. Okay. Good evening, sir. You're welcome.
Thank you. Good evening.
Good evening.
Am I being heard? Yes, thank you very much. I would like to thank the organizer for the opportunity to be sharing experience with Ironman in Nigeria. Ironman is international registry of men with advanced prostate cancer. This is essentially not a clinical trial. It was strictly an observational work that we're involved with. The project is being sponsored by PCTC, that's Prostate Cancer Clinical Trial Consortium, and Movember Foundation. The international PIs are Daniel George, Philip Kantoff, and Lauren Mushi. And this project was facilitated to Nigeria by Professor Folakemi Odedino of Mayo Clinic, Jacksonville, USA. She's a Nigerian, originally basically trained as a pharmacist, whereof to be a global scientist in prostate cancer. Iron Man has been in 15 nations to the North America, South America, Europe, and Africa. In Africa, there are 3 nations that are involved in Iron Man: Nigeria, Kenya, and South Africa. Then you also have Iron Man in Australia. There have been over 100 sites so far that have been activated. In Nigeria, I'm the PI, the national PI, and we have 4 main sites registered with the Iron Man Consortium and with the PCTC as sites in Nigeria. There's a site in University of Ilorin Teaching Hospital where I work. There's one at Federal Medical Center, Abeokuta, and 2 others at University of Maiduguri, and then Lagos State University Teaching Hospitals. In order for the experience to go widely in the nation, the CAPTC, that is Transatlantic Prostate Cancer, whose founder, Professor Dedino, brought it to Nigeria for us to be part of. She felt there should be sub-sites in Nigeria so that many more institutions in Nigeria we have this experience. It's a unique experience because we went through several training, and also the experience we've gone through so far has prepared many of these sites to participate in many other international global studies, even in clinical trials, which we are already involved with now. So as University of Ilorin Teaching Hospital sites, 2 other sites were registered under the University of Ilorin. That's the National Hospital, Abuja. We had Dr. Isia Kalawal presently as the PI for National Hospital Abuja. We have Ekiti State University Teaching Hospital. Then Federal Medical Center had Obafemi Awolowo University Teaching Hospital as the sub-site. And University of Maiduguri had Ahmadu Bello University Teaching Hospital and the Federal Medical Center be there. Then Lagos State University as a site had 2 sub-sites. They have Lagos University Teaching Hospital Booth. Dr. Antonia Showme is a PI there. She's a clinical oncologist. And then the only private cancer center that was involved is Lakeshore. We have Dr. Mutiu Jimo. So what are the objectives of Iron Man? The first one is to describe the practice pattern of therapeutic agents in the treatment of advanced prostate cancer internationally. 2, to assess whether specific treatment patterns are associated with clinically significant adverse events and evaluate potential interactions, concomitant medications, or demographic factors. Thirdly, to identify associations between treatment sequences or combinations and overall survival. 4, define the patient experience of men with advanced prostate cancer and identify unmet needs in their treatment. 5, to identify clinical and molecular disease subtypes that predict response to individual treatment, combination, or sequences. So to achieve this, the Iron Man protocol is for each patient for 5 years. When a patient is recruited, we follow up this patient for 5 years, but we know in Nigeria, in a study published in 2015, 64% of patients with advanced prostate cancer die within the 2 years of treatment. So, but we are trying to look at this. At the end of the 5 years of follow-up, we'll be able to give a figure whether this 64% is still what we have. So for these 5 years, for each patient at baseline, the informed consent is obtained, medical oncological information, demographic epidemiological factor, blood samples are taken, and the performance status of the patient is recorded, physician questionnaires, patient-reported outcome, cancer treatments, and laboratory tests that the patient had in the past. The concomitant medications are recorded and clinical significant adverse events. So this patient are seen every 3 months. And for the first year, blood sample is taken. However, anytime when there is a change in the treatment of the patient or an intervention, blood samples are taken for the test. Otherwise, the blood samples are taken every year. And this is repeated for 5 years for each patient. Of course, we lost patients along the line. So by May 2024, the Ironman started in 2017, and the Nigerian site joined in 2021. But by 2021, the Ironman site, the Nigerian site joined second quarter of 2021. This is the accrual. The intention of Ironman is to recruit at least 5,000 patients globally. And the sites were recruiting patients. By May 2024, our Nigerian site, we started about 4 years after sites in the US and in other places, were recorded very good, we have good accrual. By this time, the first Nigerian site was number 4. That's the University of Maiduguri, and has accrued 127 patients even to the registry, and followed by my hospital, 123. And this is not surprising because we have a large population of prostate cancer. And that opens up, it makes the Nigerian institution a worthy institution to be involved in clinical trials related to prostate cancer. And this is also evident. By August 2025, the global status shows that the Nigerian sites have overtaken even sites in the US that started recruiting like 4 years before we joined. At the end of August 2025, we have 221 from Ilorin alone. And I must report here that the sites have stopped recruiting as of about May this year because they felt we have attained our quota. Because already we have about 500 patients recruited from Nigeria, which is about 1/10 of the 5,000 required globally. So what we are doing now is we are following up the patients that are still on the study. And you can see Nigerian site is number 1, 221, haven't stopped recruitment. Then we have number 3 again, Maiduguri, 162. We have at least over 100 sites. So some Nigerian sites are not listed in this place. So far by August 2024, we have 2025, we have 524 patients that have been recruited. Like I mentioned, there are 4 main sites in Nigeria: the University of Ilorin, and registering under University of Ilorin is Ekiti State University and National Hospital Abuja. So these 221 recorded from University of Ilorin Teaching Hospital are actually patients recorded from those 3 institutions. Likewise, for University of Maiduguri, 162, you have it's patient recorded from mainly University of Maiduguri Teaching Hospital and Ahmadu Bello Teaching Hospital. Unfortunately, Federal Medical Center, BIDA, could not recruit a single patient throughout the trial. And Federal Medical Center, Abeokuta, also recruited 80. Lagos State University Teaching Hospital, we comprise of which had sites like Lagos University Teaching Hospital and the Lakeshore Cancer Center, recruited 61 patients. So we use the Medidata Array and the True NTA for this study as the database. And what we did was collect the patient date of birth, organization, clinic, consent status, consent date, and disease state. So what is the importance of Ironman in Nigeria? Yeah, it is important to note that prostate cancer is a major health problem in Nigeria. And over the years, it has gained prominence and in terms of incidence and disease mortality. Majority of prostate cancer in Nigeria present in advanced stages. That no wonder we're able to outspace centers that started before us, because majority of our patients in some study report up to 85% or even more that have advanced prostate cancer. When you bring all the prostate cancer, it also, this Ironman also gives us the opportunity to describe the pattern of therapeutic agents for the treatment of advanced prostate cancer in Nigeria. There are guidelines, but the truth of the matter is many centers do not follow the international guidelines because of the practicability of managing these patients in Nigeria. Essentially, one of these reasons is cost, because not many patients can afford some of the medications which are listed in the guidelines. And it also helps us to describe behavioral patterns of Nigerian patients across the country, how this impact on presentation, stages of presentation, response to treatment, and adverse events, and also to identify association between treatment sequence or combination and overall survival. We'll be able to identify clinical and molecular disease subtypes and predict response to individual treatment combination or sequences among Nigerian men, because we are doing genomic studies even on this patient also. And this Ironman study in Nigeria, because of our performance, it opened up Nigerian sites to feature collaboration, clinical trials, and funding, that the sites in Nigeria are mature enough to participate in clinical trials because of performance even in this Ironman study. What are the gains that we have gained so far in Ironman project in Nigeria? One, it has helped us to raise a crop of multidisciplinary clinical researchers working in many institutions across Nigeria. Some of the PIs are urologists, we have clinical oncologists, we have pathologists, and in this Ironman studies. So it helps us to raise researchers, multidisciplinary researchers in many institutions across Nigeria. It also made us that before these Ironman studies, there were many researchers working in various places who did not even know each other in Nigeria, but this has brought them together and made them to be able to work together even with subsequent studies. It has also helped us to strengthen the building of multi-center national collaboration, which the transatlantic prostate cancer has set out to develop. So the Ironman has helped to build multi-center national collaborations. Now we have collaborators across many sites. If there's any study clinical trial today, you could close your eyes and call researchers, faithful researchers across the geopolitical zone of Nigeria. And this is what Ironman has done for us here. It has also helped us to audit research infrastructures across the country and help us to raise awareness of the shortfall in infrastructures necessary to undertake international style projects. When we started Ironman project, every site needed a cold centrifuge and also to have a minus 80 degrees. It was surprising that many centers did not have clear access to minus 80 degrees, nor have cold centrifuge. But through the workings of the Ironman, we were able to record these infrastructural deficits and see how we can mitigate against them. Then researchers have also learned to work around deficit. They could liaise with others and see how they could work. In my own center, we had to work with molecular labs in my center, even to be able to, because my department doesn't have a minus 80 degree freezer, but working through Ironman, and because this became a necessity, we had to look inward and be able to find out that there are a number of minus 80 degree freezers within the hospital system, not necessarily related to where I work on a daily basis, but we're able to work around deficit to achieve results. And researchers, I'm a urologist, practically just treating patients, but over time, we're working with transatlantic prostate cancer, especially with the Iron Man project. I've become a better researcher. I'm able to do many more research works along with my clinical practice. So the other gains, one, they also help us to create international linkages or networking for Nigerian researchers. Now we are known to be, and we have had offers to participate in other studies because of our performance with the Iron Man project in Nigeria. The Iron Man has also provided support to upgrade available research infrastructures in institutions. There are many centers that did not have minus 80 degrees, co-centrifuge, and other things at the start of the Iron Man project. Presently, virtually every site has a minus 80-degree freezer, has a centrifuge and other facilities. And in fact, a site has, one of the sites in Capital C has solar panel to maintain their freezer. So this is one of the gains that we have gotten from Iron Man. It has also given us opportunity to have molecular typing of prostate cancer in Nigeria. This is being done with the costs imputed in the research in the grant bin. And also, we've been able to define the Nigerian patient experience with advanced prostate cancer and also to identify unmet needs in the treatment of prostate cancer. The project will also help us to identify, we mentioned this, and it has also helped to advocate for patients with prostate cancer in Nigeria, because studying all about prostate cancer, their behavioral, the unmet needs of prostate cancer will be better advocates. Yes, because we needed to translate the informed consent and the protocol into Nigerian languages, 4 Nigerian languages, English, we had Yoruba, Igbo, Hausa, and the Nigerian Pidgin language. We needed to engage Nigerian linguists, professional linguists in Nigeria. This has helped them. We have helped to develop Nigerian linguists who help with this translation. They were made to put in their best, and they produce excellent translations, which they were not exposed to in the past. So this Iron Man has been a very good gain, even for the linguists who are not. So we have also made some publications with the Iron Man. We are still on about the 3rd year. We started 2021 or so, about our 3rd, 4th year. We're still on. There are a number of publications that have been made, clinical research on low research countries, lesson learned from the International Registry of Men with Advanced Prostate Cancer. So this has been published. And we also have one, this is an international one for the global Iron Man. Nigerian researchers are part of these global publications. Iron Man, a novel international registry of men with advanced prostate cancer. And then finally, we're also talking about socioeconomic factors involved in the survival of a patient with prostate cancer. So we have this marital living arrangement and surviving among individuals with advanced prostate cancer in the International Registry for Men with Advanced Prostate Cancer. So I wish to thank Professor Folakemi Odedina, who he has been a great mentor and helped to facilitate all of this and helped to expand the lives of many Nigerian researchers across the various states of Nigeria. Professor Rotim in the PCTC, the sponsor, the Mayo Clinic, and the all Capital sites, all researchers from all sites, and the National Research Coordinator, Mr. Toit too. Thank you very much for your attention.
Thank you so much, Prof. Thank you so much. I'm sure many of us here from the SparkNet sites, some of what Prof presented resonates with us in terms of the importance of registry and the gains that we can see. So quickly, we'll go to our last presentation. That presentation also will last just 15 minutes or less. And I want to invite Dr. Adedayo Joseph. Please, the team, can we make her a co-host so that she can do her presentation? Dr. Adedayo Joseph, you're most welcome.
Unmute, please.
Apologies, I didn't realize that I was muted. So I was saying good evening, everybody. Thank you for the invitation to speak today, to share our work with you. And until now, I'm going to share my screen. Okay, so thank you very much. Please confirm that you can see my screen.
Yes, we
Yes,
can.
we can see your screen.
Thank you. Okay, so good evening, everybody. My name is Adidaya Joseph. I'm a clinical oncologist at the MedServ Youth Cancer Center in the Lagos University Teaching Hospital. So the Youth Cancer Center is a private-public partnership between Youth and MedServ, Nigerian, the NSI, the Nigerian Sovereign Investment Authority Medical Services. So that's just a brief introduction. So just to talk about the Hypo Africa today, and I'll go into details about the trial, and as I go along, talk about some of the impacts that it has had on us. So, but the Hypo Africa trial is a multi-institution transcontinental clinical trial consortium that's made up of investigators from across continents, and that's why we're calling it a consortium. And it is the Hypo Africa it's the short form of hypofractionated radiotherapy for prostate cancer in Africa. But just as an introduction, everybody knows that Sub-Saharan Africa is going through a cancer crisis. Between a combination of raised awareness, increased diagnostic capacity, the incidence of cancer and the diagnosis of cancer is rising, and rising dramatically compared to the rest of the entire world. And so, you know, it has a number of things have emerged from that. So one of it is understanding and recognizing that we do have a crisis on our hands. But another thing is the emergence of research, the emergence of clinical capacity, and the understanding now that Africa is sort of the next frontier in cancer biology and cancer science. So radiotherapy is indicated in up to 50% of cancer patients over the course of treatment. At some point, every patient diagnosed with cancer will need, at least half of patients diagnosed will need radiotherapy, either as definitive treatment or as palliation of symptoms like pain or bleeding or metastatic sites. And currently, there's a significant deficit of radiotherapy capacity in terms of machines, expertise, and so forth on the continent. As of now, Africa is estimated to need about 5,000 more radiotherapy machines in order to meet the standards set by the International Atomic Energy Agency, which is like the agency that determines how many machines you should have across the world. So the recommendation is that you should have a minimum of 1 machine to a million population, but many African countries do not meet that requirement, and some African countries don't even have a radiotherapy machine at all. And the deficits are in multiple domains. So there's the machine deficit, but there's also a lack of experts and expertise. And when I say experts, I mean in terms of numbers. So there's, as we all know, a knowledge exodus out of the continent of Africa. There's people migrating out of medical practice, but also people migrating out of the continent entirely, out of, you know, African countries. There's also, because of that, then a shortage of expertise. And that is a combination of things. So we don't have training programs, enough training programs in the continent. And we are losing those that have been trained to other climates so that there's no trainers left to train the next generations. There's also deficits of maintenance. We have a lot of downtime that are connected to issues such as power. So when you don't have stable power, radiation machines will break down. And these machines are very expensive to purchase, very expensive to maintain, very expensive to repair. And that will then contribute to further deficits. There's also difficulties of geographic access. So using Nigeria as an example, the landmass of Nigeria is quite huge. And then you have, you know, like the greatest concentration of machines in urban sites like Lagos, like Ibadan, like Abuja. So for many people who do not live in those urban sites, there is limited access to radiotherapy for them. And finally, there's financial access. As we all know, 90% of treatment costs are funded out of pocket in Nigeria. And so what that means is that there are many people who, when they fall sick, cannot afford access to radiotherapy. And radiotherapy is expensive everywhere in the world. If you were going to do a world comparison, it is actually relatively inexpensive in Nigeria. But that is relative to the world, not relative to average household income. When you compare it to average household income, it is inaccessible to over 70% of the Nigerian population. So just to talk specifically about the trial now, hypofractionated radiotherapy. So as a background, radiotherapy is like surgery. You are trying to cure a tumor, right? Without using systemic therapy, without giving medication. You're using a different means other than medication. The difference between radiation and surgery, and I will go further in that comparison as I go later on, is that with radiation therapy, we're not going in and cutting open the person. So the first thing is that it's not invasive. We don't want to cut them open. We want to achieve the same results you would achieve with surgery without an invasive procedure. The second thing is that you don't do it at one time. So with surgery, you anesthetize someone, you put them on an operating table, you go cut out the tumor and you're done. With radiation, we're going to be giving the radiation little by little. And the rationale for this is that, and this is how I explain it to patients when I'm explaining, to say that the truth is that if I give 100 gray of radiation to this breast tumor today, in this moment, I will eradicate the tumor, absolutely will, but I will also eradicate the person. And so because we cannot give that amount of dose at one time, we break it up and give it little by little by little, with the understanding of tumor biology being that tumor cells are immature and less able to recover from what we call sublethal damage. So every time I give a dose of radiation, I'm causing sublethal damage. The cancer cells are immature, they cannot recover, but the normal cells can recover. So you give a little bit of dose, there's some damage to all the cells that radiotherapy dose hits. The cancer cells do not recover, the normal cells recover. And then you give another one and another one, and it can accumulate like that over time to be able to eradicate or at least halt the progression of that tumor. And at the same time, the normal cells are expected to recover because the capacity to recover more than cancer cells. So that is sort of the that's a very simplified explanation of why we fractionate. So what we give, the daily doses that we give are called fractions. And so this is a rationale for why we fractionate in radiotherapy. Now, the standard fractionation over time, historically, has been conventional fractionation, where we give 1.8 to 2 gray per fraction over several different treatments. Using prostate cancer as an example, in the past, we would treat prostate cancer for 40 fractions. Fractions are given Monday to Friday, no treatments on weekends, no Saturdays and Sundays. So giving someone 40 fractions of treatment means that he's going to be coming to your center every day for treatment for 2 months. That is a significant amount of time to dedicate to traveling, let's say from Ikorodu to Luth every single day to get treatment every single day. Many people will lose a lot of income from their businesses, will not be able to hold on to their jobs. The cost of transportation, the loss of childcare, the impact of that amount of time dedicated to treatment is significant. And so what hypofractionated radiotherapy does is that it tries to reduce the number of times the patient needs to come to the hospital. So instead of 40 fractions, we're giving 20, but we're giving a higher dose every single time. So instead of giving 2 gray and then treating 40 times, now I'm giving 3 gray and treating 20 times. And even though the final dose is not the same, there's something called biologically equivalent dose. And with the increase in fraction size, the BED changes as well. So there's biological factors that are factored into the dose that is given every time. So that is a basic rationale for hypofractionation. Now, the Hypo-Africa study has had similar studies done in other countries, a hypo trial, CHIP trial. In fact, the CHIP trial is very popular. And based on that, we wanted to demonstrate feasibility of doing this regimen in Africa. It's particularly significant for Africa for a number of reasons. So like I said, we don't have enough machines. We don't have enough experts. I can speak for myself to say I am overworked, I am tired and exhausted. And that goes for the same for medical physicists, same for radiation therapists who are also involved in this process. So if we can get someone from the beginning to the end of treatment faster, that works best for everybody. It works best for the patient, for their family, but it also works for the system as well. We want to get this person off the machine so that we can welcome another person for treatment. It provides access to people. It limits the waiting time that they have to wait for a space to become available and then get on the machine. It is also economically sound. It's economically sound for the patient. It costs less transportation. They spend less time away from their place of business. It is also economically sound for the system. The resources that we put into treating someone 40 times is much more than what you put into treating someone 20 times. The final benefit is clinical. There's something in radiation that we call shifts. we call treatment migration, intrafaction shift, meaning that when we give radiation, we want to position a patient exactly the same way every single time, because you have to make sure that every single time you treat, you are hitting the target and you're not hitting the normal structures that surround the target. Now, if you think about it, it is just reasonable and makes sense that it will be easier for you to hit the target perfectly every single time if you're treating 20 times than if you're treating 40 times. It is almost impossible to get to the end of 40 fractions and not have had some migration of your target, of either your target or your positioning or something. So ultimately, there is a thought that with shorter fractions, your treatment becomes more accurate. And that brings me to talk about something that is not part of this, but I'll just mention it slightly because I used the surgery example to say that there is something called stereotactic radiosurgery. And it is called radiosurgery because it is done in one single treatment. And you are giving your full dose at that one time, but you're giving it through multiple beams so that the amount of dose that is passing through every part of the normal structures is minimal, but they all converge in one place to concentrate that full dose. So just mentioning that, by the way, because I used the surgery example as a comparison. So talking about prostate cancer, why it is relevant to do studies on prostate cancer, the previous speaker has covered everything. Prof has said everything that I can think of to say in terms of that. But just to mention that, it's the leading cause of cancer-related death in men in sub-Saharan Africa. African prostate cancer is different from Caucasian prostate cancer. African men develop prostate cancer at a younger age. They tend to have more aggressive disease. They have higher PSA values for disease stage. We see prostate cancer patients who come with prostate values that, PSA values that our international colleagues say, oh, is he still walking? Or you see metastasis in the spine where the entire spine is covered in mets. And, your colleagues are shocked to find out that he walked in by himself, he's still walking. So the kind of prostate cancer that we see is different. The biology of prostate cancer in African men, and this has been documented, is different. And the one thing that we also know is that mortality is higher. The African man is more likely to die from prostate cancer, even when he lives in a high income country than the Caucasian, for multiple different reasons, biologic and social. So it is important to study prostate cancer in African men. And that is why we couldn't just take data from the HIPRO and the CHIP trials and just paste it on African men. We needed to do the study in African men to confirm that the outcomes we would have in our population will be the same as those seen in Caucasian populations, understanding that the type of prostate cancer they have is different. So this is one of the papers that we've published with regards to hyperfractionated radiation among the team. But just to talk about Hyper-Africa specifically, like I said, it's a multi-center study. It is prospective, non-randomized. It's impossible to randomize because there's no way to pretend you're treating someone 40 times when you're treating them 20 times. So the people who are being treated 20 times know that they're getting 20 treatments. So there's just no way to randomize on that type of trial. And the real goal of the study is to assess non-inferiority. We want to prove that giving fewer fractions will not compromise outcomes and will not worsen side effects. And that is the summary of everything. And so 3 sites were involved. The NSI Youth Cancer Center, like I said, which has now rebranded to the MedServe Youth Cancer Center in Lagos. The Nkosi Albert Lutuli IALCH in Durban, South Africa, and Ocean Road Cancer Institute in Dar es Salaam, Tanzania. And with future iterations, we now have a center from Uganda, the Uganda Cancer Institute, joining the study. So 182 participants were recruited. We divided into 2. Low and intermediate risk patients had 60 gray. High risk patients had just 3.1 gray. 0.1 was added to each fraction of their 20 to give 62 gray. And the primary endpoints were to assess, gastrointestinal and genitourinary toxicity and cumulative late toxicity later on. So these are early, this is early data, and I'm not going to speak extensively about this because we haven't published yet. But however, even from preliminary data, we're beginning to see that our toxicity scores are similar to those that have been done in previous studies. And that is sort of, tending towards the fact that hyperfractionation is reasonable and safe and feasible in Africa. These are the principal investigators on the trial, Professor Twali Bongoma in Tanzania, myself in Lagos, Nigeria, Abba Malum in South Africa. And like I said, the Uganda Cancer Institute is joining us with Solomon Kibude from Uganda. Luca Nkochi, Prof. Luca Nkochi at Erasmus Medical Center in Netherlands is, and Will are the main PIs of the study. Wilfred Ingwa is a radiation oncology medical physicist working in the USA. And so just to talk about the team structure. So like I said, those are the PIs, but there's many people who work on this Hyper-Africa team. Our medical physicists, the data team, the radiation therapists, the nurses. For each site, there's multiple people. And over time, the group has grown and expanded. I'm sure we're almost 40 or 50 people in that group now, and multiple other studies are coming out of it. We have weekly meetings every single week, every Saturday, and we've done that for several years. I think 3 years now, we've had meetings every single Saturday. And then every now and then there's ad hoc meetings and discussions on WhatsApp and all of that. And then mentorship programs have come out of this. So there's a global medical physics mentorship program that has come out of this. Oh, and talking about the team, I just needed to acknowledge, and I will acknowledge her again, Katie Graff, who is the secretariat of the consortium. So Katie keeps everybody on point in meetings. She keeps documents together. She organizes the team structure. I can't say enough about the support that Katie and BVGH offer. So I was talking about mentorship. So there's a medical physics mentorship that emerged from the trial just because as we started, it became evident that there were certain gaps in medical physics structure on the continent, and a very strong medical physics research group has emerged out of that. We've also done a breast cancer digital course at some point, as well as a number of other courses at different points in time. As we go along, the Hyper-Africa team is working on some other upcoming trials, including immunotherapy. We want to go to ultra hypofractionation. So instead of doing 20 fractions of treatment, we will now be doing 7 fractions for prostate cancer. We're also looking at ultra hypofractionation for breast cancer, which is similar to something called the fast forward trial, looking at cervical cancer as well. And then there's multiple different projects, the DUAS project, which is going on, the ENSURE project, and things like that, all dedicated to radiotherapy, global radiotherapy. And as I wrap up, I just want to talk about mentorship. So when the clinical trial started, this was my first clinical trial, and I started off as a principal investigator, which was a significant challenge for me. But I find that the challenge was more mental than real, meaning that it was more out of my own fear of my inadequacy, because I mean, I'd never done a trial before, so to do your first one as PI was daunting. And so I'm very grateful for a team of people who were extremely supportive. And, so for me, I learned how to mentor from my Hyper-Africa team. And it starts with respect. It starts with knowing that even if you've not done one before, and this, I remember Will saying specifically to me that you may not have done one before, but it's not for lack of knowledge. It's not because you don't have the intelligence to do it. You just haven't done it. And when you've done this one, then you would have done it. And it's really that simple. And, so that understanding, that looking at another person, regardless of how junior they are to you, and understanding that the person brings value, even if they don't have the experience that you have, is the foundation of good mentorship. The next thing I would say is collaboration, truly working together. So there's collaboration and there's control. And I have been close to people who are in, group studies where what you see is an exertion of control rather than a true collaboration. And it's one of the things I'm grateful for the Hyper-Africa Consortium is it is truly a collaborative team. The next thing is inclusivity. This is not we're not a cool club. Everybody is welcome. Everybody brings value. Everybody is bringing something, and everybody else that is there already is open to learning from whoever is new and is joining the group. So inclusivity is very important and very celebrated. Consistency. So like I said, we've had meetings every single Saturday, and I find that when I have to miss a meeting, I feel so guilty because of all the other people who are dedicating that time. There's some people who have never missed a meeting, and no matter what is going on, the child is getting married or, things like that, they're still showing up every single time. That level of consistency is admirable and is one of the things that I've learned about being a mentor, because it then makes it difficult for the person who is being mentored to not show up. And then commitment, and finally, determination. And so as I wrap up, again, I just want to acknowledge Katie Graff of BVGH, because the Hyper-Africa team will be falling apart without her. So Katie, thank you very much for all your efforts, and thank you everyone for listening today.
Thank you so much, Dr. Adedayo. Joseph. Thank you. It's good to see you sharing practical experience, PI of a clinical trial from the very base. So quickly, time has gone, but we'll just take one or two questions. Please, can the IT team look at the chat and see if there are any questions there? We'll just take one or two questions, and then we'll invite Professor Nodu to close us. But just to say that for the recordings, where the presenters are not there live, please, we're going to have the recordings of all these, and every question that people have posed, we are going to answer them. And the good thing is we all have your emails, and in subsequent sessions, we'll provide answers that presenters have given us when they don't have live presentations this way. So thank you. Thank you. So can we hear from the admin team, one, two questions that people might have posted in the chat?
Yeah, we just have one. How do we maintain an audit trail?
Okay, so Dr. Shea is still here. Dr. Yango is still here. Please, who wants to take on that? How do we maintain?
How do we maintain an audit trail?
Is Dr. Kinshote here? Is he here? Okay, he is. Please, Dr. Kinshote, could you handle that? I'm not sure he can hear me. What of Dr. Anyango? Is he still on the call? I don't think he is.
Is
I see
Frida
Dr.
on the call? Frida? Anybody on the call? Any of the faculty?
Is there any,
on the
yes,
call?
any of the Archivia team on the call?
Okay, so maybe we'll take the next one, and then we intend to draw up an frequently answered questions for this course. And all the materials for this course will be at the Nesta website, so that people can log in and view them. Also, the frequently answered questions the answers will be at the SESTA site, so that people can go in there and then review the presentations and the rest of the information that we have.
Okay, so please, the admin team, can you just one more question from the chat? I think you had about 3. Can you just read out one more? Let's see if we can tackle that before we close. Oh, I can see that Dr. Yango says he's on the call. So if he could help us.
Yes, I was unable to unmute, but now I've been able to unmute.
Okay. So the question is, how do you ensure an audit trail?
So how do you ensure an audit trail? So that's a good question. So it will depend on if you are utilizing paper source documents or electronic source documents. So if you are utilizing paper source documents, which will be most of the times, then it means that for any entry that you make, if you make changes to that, Then you need to initial and date so that we're able to link who made that change and when was it made. And if the reasons for the change is not obvious, then there's a requirement that you put a small explanatory note to say what was the rationale for making that change. Now, if you are utilizing electronic medical records or the EDC system, then it should be designed in such a way that one is able to capture in the details of who is the site staff who has logged in. And in the event that you need to make any changes, one, it will not completely obliterate the original entries, which is also the expectation for paper source. But then again, electronically, you should be able to capture what is the reason for the change. So was it a data error? Was it a new entry? And things like that. Then for electronic systems, it will keep it in the background. But for paper source, you will need to document all that just beside where you made the original entry without completely obscuring the original entry.
Thank you very much, Dr. Yango. Please, we've overshot our time. So please, I want to plead with everyone that we'll try to address all the questions that might have popped up in the chat so that we can close. Please, Professor Nodu, the director of Ancestor, please can you close us?
Okay.
Thank
Yeah, thank you very much. This has been another exciting time of learning, for me personally, and I'm sure, for other people. I didn't know. I mean, Professor Dedino is a friend, but I didn't know to the extent of what her consortium is doing. And so I thank Professor Pupola, for bringing that Iron Man, a consortium, to our attention. A bit similar to what we're doing in the Sickle Pan-African Research Consortium Network in Nigeria, where we have 25 sites and we have over 10,000 patients in a disease registry that we have been following up over the past 8 years. And it is precisely because of the presence of that registry that we're now doing this course on clinical trials in order to get the 19 sites that are not currently doing clinical trials in the within the network to get them ready for clinical trials. So this has been really exciting. I thank all the presenters, from Nnamdi Okeke, who also has his own CRO organization. All the presenters. Sheye presentation was really very detailed and very practical and resonates with all of us. I thank BVGH for bringing the rest of the faculty to this, and I want to thank everyone because I saw that we were over 215 participants on the course logging in tonight. So we still have capacity, to have more people. So please, if you can get your friends to stay with you, if possible, then that will make more, it will give us opportunity to have more people benefit. But essentially, the presentations will be available, to all of us so that we can listen again. I have benefited from doing that on occasion, and I want to thank everyone. I thank Professor Maxwell, and I thank the IT crew. I thank everyone. I wish you a good night's rest until we meet next week. Thank you very much.
Thank you very much. So good night, everyone. Once again,
Good
thank
night.
you to all our presenters. Good night.
Okay.